Good day. This is Marco from AB. I'm going to talk to you today about thiols and esters optimization, especially in Sauvignon Blanc, especially during fermentation. As we know, wine aromatics can be primary, secondary, or even tertiary. So from the grapes, there would be primary, like terpenes, methoxypyrazine, the herbaceous xanol. Secondary, like the thiols that we're going to discuss today, norisoprenoids, esters, and then other aromatics that can come up with aging. As far as the thiols, we have the usual suspects here. Let me actually make this bigger. So the 4-MMP, the 3-MHA, the 3-MH. Esters can be acetate esters, the fruity and floral, and then the medium chain fatty acids of various nature, more citrusy, sometimes soapy. Talking about thiols, they are normally derived from a sulfur, including amino acid like cysteine, like glutathione. They're linked to those specific amino acids and need to be released. So the process normally in winemaking is to first actually get this sulfur containing amino acid and then trying to extract the actual thiol from its precursor. You see here the 4-MMP, the 3-MH, the 3-MHA, as I said, the usual suspect as far as thiols, with slightly different aromatics. The 4-MMP is normally more identified as box tree, as even cat pea. The 3-MH, a little more citrusy. 3-MHA, guava fruit, more tropical. If we have thiols in red grapes, certainly on Grenache, for example, we can have blackcurrant aromas, so slightly different in a different matrix. These precursors that we were talking about, the sulfur-containing amino acids, where can be found? Mostly in the skin of the grapes. We see here the cysteine 3-MH precursor, it's mostly in the skin. The glutathione 3-MH, again in the skin. The glutathione 4-MMP in the skin. The cysteine 4-MMP, skin and pulp. To get them off from the precursors, what is needed is an enzyme. We call it CSL, a carbon sulfur lyase, that normally is released by the yeast, is part of the metabolic process of the yeast. Certain yeast have it, more enhanced, is part of their genetic encoding. Other yeast don't have it, so that's why there are yeast that are indicated for Sauvignon Blanc, and some of the yeast we're gonna discuss today from the AEB collection. This release is actually gonna be good for, as we see, the 3-MH and the 4-MMP. There is another thiol that we have mentioned, which is the 3-MHA. That is just derived from the acetylation of 3-MH. Also done by the yeast. The yeast that can do it needs to have a specific gene to be able to have this particular transaminase that allows the transformation of this 3-MH, which is alcohol, into the ester, the 3-MHA. So here we're going to discuss what are the enological practices that allow us to have a better expression of these thiols. As we mentioned, the yeast strain is very important, needs to have at least these two genes encoded. So the one for extracting from the precursor, the carbon sulfur lyase, but also the specific transaminase if we want to have more variety and transform the 3-MH in 3-MHA. Nutrients, as we're going to see in the course of this presentation, are also important because they can be a substrate for the yeast to get more precursors. Enzyme, as we mentioned, enzymes are important in the metabolism of the yeast. We can help with certain enzyme the metabolism of the yeast, but also there are enzymes that can help the extraction from the skins of the precursors, and we have seen how many precursors we have on the skins. Oxygen management, because these components are all sulfur based, and as we know, sulfur based aromatics are very easy to oxidize. A note at the bottom, for everything to work out to perfection, the temperature shouldn't be too low. We know that white wines sometimes are fermented well below 16 Celsius. That would be just below 60 Fahrenheit. But at that temperature, enzymes don't have much of an action. So as we mentioned, the enzymatic activity is rather important. So we do want a temperature that is at least 16 Celsius, at least 58 Fahrenheit, even better if it goes to 20 Celsius. The temperature needs to be low during storage to preserve eventually these thiols. So we discussed the yeast as an important factor in generating thiols. We are not talking about yeast that are, genetically modified to obtain our result. There is a lot of speculation from the natural wine producers that certain wines are made, are almost manipulated. In this case, we're just trying to find an yeast that does what we like, and we try to make that dominant. The only, in a way, manipulation that we can do in a lab without doing GMO, which is not what we do, is an hybridization. Basically, have a sexual mating of two strains that have characteristics that we like, trying to enhance the specific characteristic that we want. So in order to do so, we need to have a sexual mating of the yeast, which is not something that is done normally. Normally the yeast would bud, but in a certain condition of stress, the yeast would sporulate, and we can actually induce a sexual mating. So just to give you an idea of how these procedures work, you choose the yeast that you like. In this case, for example, we have a couple of yeast represented here from our collection. The Fermol PB 2033, the Fermol Candy, very aromatic yeast, normally used for rosé actually. We can make that, have an hybrid and more hybrids, and then go select. The first kind of selection that we do nowadays is low H2S production. We also like to have a killer phenotype to have dominance in the fermentation, making sure that our strain kills competitors. And then once we select that in the lab, in the Petri dish, we take those selected strains for industrial trials. The strains are big number originally. We started with 28 candidates, and our first parameter of selection in the industrial trial was the fermentation efficiency, so low production OVA. Out of 28, we restricted to 11 candidates. Then we went on in our French plant, actually you see the notes here in French, to a sensory evaluation. We were looking for citrusy, as you see here, agrume, and other characteristics that we decided were positive that made us select 6 strains out of the 11. Third parameter of selection, ability to ferment under nutrition stress condition. We want to make sure we don't need to have IEN, because what we're also going to see in the rest of the presentation, having more amino acid and less the ammonium phosphate in our nutrient is key, but that's going to result in lower EN addition. And so we need strains that are able to ferment with low yeast available nitrogen. So third parameter of selection. Fourth parameter of selection is again sensory analysis, and we selected 4 strains out of 5 candidates. And then we focused on Sauvignon Blanc. Focusing on Sauvignon Blanc means finally going after the thiols. And here we see how the thiols are outlined mainly on the 4-MMP, the 3-MHA, and the 3-MH. We have a reference yeast here that is used a lot on Sauvignon Blanc that resulted very high in the typical Cat P box tree aroma, very expected, let's say, in a Sauvignon Blanc. We were looking for something different, something more modern, tropical fruit, 3-MHA, citrusy, like its alcohol, so the 3-MH. And we got some results that were very interesting with some of these hybrids, the 101, the 121, and the 171. The 101 that was higher in the citrusy was then called Fermalt Lime. So high producers are the 3-MH, so the thiol that is reminiscent of grapefruit mostly. We add more tropical aromatics, more guava, with the yeast called Fermalt Fleur and Fermalt Tropical. So these 3 strains with this name were adopted and commercialized by AEB. In the same research, there was a 4th yeast, as you remember in the last slide of the selection. This yeast being a little more extractive, and so giving a little more depth to the wine, was selected for red that need to be fresh, aromatic, sold within the 2 years. So with a very vibrant fruit, even for rosé. We're talking Grenache, which is a variety with very pronounced thiols that give that particular fruity red cassis note, blackcurrant. High potential for this blackcurrant is also in Pinot Noir, Cab, and Merlot. Young, of course, meant to be drunk in the next 2 or 3 years. So the Fermalt red fruit was developed specifically for this kind of wines. Another yeast that is part of our catalog and that we recommend for Sauvignon Blanc is a non-Saccharomyces, Torulaspora durbrechi, which in the AB collection is called Levullia torula. So, as we know, in the microbial population dynamics in wine, non-Saccharomyces proliferate very well at early stages when the alcohol is below, let's say, 9%. And Torulaspora durbrechi is no exception. In these few days, winemakers that only inoculate immediately develop their complexity. Unfortunately, sometimes their volatile acidity. It happens in nature, and we have gone after this complexity that can be developed by this native strains, and over the years developed about 10 commercial strains that are available in the market for non-Saccharomyces, which is a very low number compared to the 300 commercial strains of Saccharomyces cerevisiae that are used normally to ferment wines. There are also 50 commercial strains of lactobacillus, of Enococcus ani and lactobacillus. So, as you can tell, non-Saccharomyces species are very minimal. We, in our collection now, sell mainly 3, the Levullia torula, which is Torulaspora durbrechi, the Levullia alcumeno, which is Lachance thermotolerant, which has the characteristic of converting part of the carbon from the sugar in lactic acid, so not in alcohol. So we're going to have less alcohol as a balance and higher lactic acid, so lower pH. So it's a very good yeast in warm climate. We also use it for sour beer, very successful in that industry. Levulia pulcherrima, which is Metznocovia pulcherrima strain. It's normally used for bioprotection because releases pulcherrimic acid, which is very active against lactobacillus and Brettanomyces. So we have a product that is called Prima Flora, which is supposed to use even on the grapes if you can, but definitely the earlier as you can, in the press at the crusher, and that's something that can substitute SO2. Going back to Levulia torula, so the Torulospora tulbrechi, what does it do for us when inoculated in the early stages of fermentation? It will contribute positively to the organoleptic complexity of the wine. So when those natural wine I just mentioned before come out good, because sometimes they'll come out good, their main feature that makes them so intriguing is the complexity of the aromatics. And that's exactly what Levulia torula will give you. That complexity without the funkiness, because it has a very low production of volatile acidity. It also has the characteristic of releasing polysaccharides and amino acid. That means a very easy lysis of the yeast, so the release of, say, the guts of the cell. So, of course, the release of polysaccharides will contribute to reduce the sensation of astringency or harshness in the mouth. And the release of amino acid can work as a substrate for nutrition for the second yeast that will be inoculated, because as we said, these yeasts are very active in the first part, but normally we want to have a sequential inoculation where this first yeast will be followed by a Saccharomyces cerevisiae to actually complete the fermentation. Also, this amino acid eventually can be used by a malolactic bacteria in case that's going to be the winemaking strategy decided. This yeast is also very high in sulfur lyase enzyme, so for the production of thiols. This is the gene that we were mentioning before that needs to be encoded in the yeast to actually free the thiols from its precursor, from the glycine and the glutathione, from the cysteine and the glutathione. Where do we recommend using the Torulospora dulbrechi, the Levulia torula? Basically, we have seen good effect in every kind of wine: dry, sweet, white, red, rosé. Obviously, mainly where you want this kind of complexity of thiols. Sauvignon Blanc is one of those. But also in reds, we have had good results in Pinot Noir. Of course, Grenache with blackcurrant and strawberries are the main descriptor normally. And in all our trials, we developed three main protocols. Well, if you want a sweet wine, just let the Levulia torula ferment through. It normally doesn't pass the 9% alcohol level. In white wine, you want a clean fermentation. So sequential inoculation is the best way to make sure the fermentation is always going and is regular. So add the Levulia torula, and then after 3 days, add another fermol yeast, so a Saccharomyces cerevisiae. In red wine, you can actually co-inoculate. You're going to have a small effect of the Torulospora, and then the other fermol yeast will take over. This is a trial that we did in Sauvignon Blanc, where we compared. It was done obviously in an industrial winery that had other yeast that they were using. The X5, which is used a lot for Sauvignon Blanc, Maxi Thiols, that was used actually in the sequential inoculation with the Levulia torula. So the Levulia torula was let ferment for 3 days, and then this other yeast was subsequently added. I had the fortune actually to taste this trial, and to me, in my mind, this graph actually represents exactly what I remember tasting. It was wine, very high expression of guava, tropical, complexity in general. So, as I was saying, almost felt like tasting a natural wine without the funkiness. Uh, X5 was very grassy, like normally it is. I thought it was also very evident that for MMP, the box tree in the panel, it resulted maybe more of the other thiols, the 3MH and the 3MHA, but definitely more grapefruit, citrusy. The maxi tile wasn't that complex, it was kind of closed, but with the Torwa, it literally exploded in this kind of complexity of the nose. As far as the mouth, the length, as I was saying, the polysaccharide gave space to a lot of lingering, a long aftertaste, and intensity of the flavor that were in the nose matching in the mouth. The X5 gave this typical green, neat flavor. Going back to this slide, now that we have looked at this yeast strain, what else can we do to enhance the thiols and the esters? Because that's also what we're talking today. We can use nutrients that amplify the efficiency of the yeast in this job of creating aromatic. In fact, the same yeast strain will originate different aromas when fermenting different substrates. That's why fermenting different grapes with different yeast, with the same yeast, sorry, will give you different aromas because the substrate changes as the yeast stays the same. When we add a nutrient, we're actually adding a substrate. So we have to consider that. It's not that the nutrient will impact the aromatics so much that we're going to completely make a different wine. The nutrient will just enhance what the variety has to express. The choice of an organic nutrient is always an excellent booster for the aromatic profile. What does that mean? If we use the ammonium phosphate or the ammonium sulfates with our mineral nutrient as opposed to organic nutrient, those nutrients will actually impact the transporter that the yeast use to bring inside the cell the precursors for the aromatics. No precursors, no aromatics. So that's why an organic nutrient always gives you better expression of the aromatic. And going back to this other slide, where are the precursors and what are the precursors? Sorry, the cysteine linked 3MH, the cysteine linked 4-MMP, the glutathione link 3MH, the glutathione link 4-MMP. Well, cysteine, glutathione, those amino acids that we can actually source from a lysis of a dead yeast and add back to the fermentation. Also, we have skin containing precursor, especially for the 3MH and for the 4-MMP, and the G3MH and the G4-MMP. So all these precursors in the skin means that if we, like the first evidence actually was a while ago, but not that much in terms of enology, which has a history of over 100 years. So in the last 10 years, there were research that found evidence of these precursors in skin tannins. No surprise, since we just said that these precursors are mostly in the skin. So why don't put some skin tannins in the fermentation, peel tannins, skin tannins, to enhance the conversion of these precursors into thiols. And that's what you'll find when using Thermoplast Sauvignon. You'll find the amino acids containing sulfur, like the cysteine and the glutathione. And you'll find skin tannins also containing these precursors. Here is a little trial that we did. It shows a couple of things. It shows that when using Thermoplast Sauvignon instead of, say, DAP, your content of any thiol is always higher. It shows that if you use an yeast that is not meant to release or convert thiols, like for example this Fermol Blanc, which is an AB yeast normally used as a workhorse for its characteristic is a variety Baianus. So it's got a lot of resistance, it's a very solid fermenter, but doesn't have certain features. So it's not going to be able to ferment as well to produce these thiols in the same way that another yeast like the Fermol Tropical, which is part of the research that we saw in the first part of the presentation, and as the two enzymes encoded for the extraction of the thiols and the conversion of the 3MH into 3MHA. So we see that when fermenting with this yeast on this nutrient, you have very high level of each of the thiol. Using Fermol Blanc, even on the Thermoplast Sauvignon, yes, you're going to have some conversion, but not as high. Enzymes, amino acids, these are all important. One of the enzymes that we identify as being important for this conversion is the enzyme thiol, which is basically a carbon sulfur lyase. That enzyme that we mentioned can extract from the precursor. Amino acids for the adding more precursors. So we developed the Thermoplast Sauvignon that just we've seen, but more of these nutrients, maybe without the skin tannin from like the Thermoplast Sauvignon. Thermoplast DAP-free, it's a concoction of amino acids, yeast-derived amino acid. The Thermoplast Tropical, that is very indicated also for production of esters and has a very high content both of glutathione and cysteine. This to introduce an actual trial that was done at the University of Adelaide. You see here by this guy Federico Tondini, PhD. At the University of Adelaide, they had this system where they were able to conduct about 100 different fermentation at the same time in a small scale, lab scale, obviously, with this T-boot robot. And here are some of the results. You see that we were comparing two yeast, a control, and this Glutaferm, which is our yeast for production of thiol. This yeast, the Glutaferm, is actually a producer of glutathione. So you see when there are no addition, it actually produces higher thiols than the control. Whereas as soon as you start adding things that help the metabolism and the transformation of these thiols, this other control yeast produces more of the thiols, but produces more of the thiols, especially when this enzyme thiol is used, so this carbon sulfur lyase is used, or the Thermoplast Tropical, this nutrient with an high content of glutathione and cysteine. So this Thermoplast Tropical matches the result of the enzyme addition. This is in Sauvignon Blanc for 3MH. This is 3MH in Chardonnay, similar result. We see the Tropical at 400 mmP, 40 grams hectoliter. We see here with the enzyme, again, 3MH in Chardonnay. 3MHA in Sauvignon Blanc, very high with the Tropical, rather low with all the other trials. 3MHA concentration in Chardonnay, high with the Tropical and the enzyme. The enzyme here made a big difference. And this, for your notes, are actually the two products, the Thermoplast Tropical, so the nutrients, and the enzyme thiol, the carbon sulfur lyase enzyme. As we were mentioning before, another big component of the secondary aromatics are the esters, and consumption of amino acid has been associated with ester formation. Consumption of amino acid, as you remember, I said, it happens, all these substrates are included in the cell, especially when the ammonium phosphate is not added. So this is sort of the first key factor for having this transformation happen, using that at the right moment and in the right amount. Why amino acid are part of this pool of precursors? We can see now that through the Ehrlich mechanism, amino acids that are not used by the yeast to synthesize glutamate in their metabolic pathway to form folic acid or other amino acid, this excessive amount of amino acid is say expelled by the yeast through what is called the Ehrlich mechanism. And here you see the pathway that leads to fusel alcohol that is then acylated into an acetate ester. These are some examples, for example, the leucine becoming an isoamyl acetate, the valine becoming isobutyl acetate, and so forth. Again, we're not trying to manipulate wines. Here is the yeast that is trying to do something for itself. First of all, because in the pathway it gets to an alcohol, and the alcohol is always a little toxic, right, for microbes, it likes to do a detoxification mechanism where the alcohol becomes an ester, which is less toxic. Also, once it has these esters that are attracting insects, the yeast is sure to have a vector to travel easier. The insect is going to touch the wine, the fermented must, and is going to carry some of the yeast somewhere else. This is a result of a trial that we did against a couple of competitors' yeast, with the yeast that were selected, remember, for thiol conversion. And what we found is that the formol lime forms more of this fatty acid derived esters, more fresh, more citrusy, let's say. The formol fleur, so the formol tropical, in this study we represent only the formol fleur, is more capable, let's say, to produce acetate esters derived from the degradation of the amino acid, so more in the red fruit, tropical, pineapple, banana kind of aromatics. So very high, as you can see, certain numbers are particularly high for this two yeast. So the Thermoplast tropical that we saw before being a good substrate for forming more thiols because it contains that glutathione and it contains that cysteine substrate, also has a good amount of amino acid to produce tropical esters. So this is why it's called tropical. AV has about 30 different nutrients that are all studied, starting from grape varietals that have certain characteristics. The Thermoplast tropical actually is derived by studying this variety here that is called Anton Vaz, which is a sort of an obscure Portuguese variety with a very pronounced pineapple aromatic. But then AB also has, for example, the Thermoplast Prosecco. Guess which one is the grape that is scanned to understand the amino acidic profile of that grape and transport that in the nutrient. The Glera variety, which is used to make Prosecco, and so forth. As I said, we have about 30 of those. We have the Thermoplast floral from Gruner Veltliner, for example. So now that we looked into the nutrients, after looking into the yeast, let's talk about enzymes. Actually, we already talked about the enzyme. Sorry, we talked about the enzyme thiol. We haven't talked about the pectinase. I don't have a slide for that, but pectinase are very useful because, especially if you have a little cold maceration in whites, can help to extract more of the precursors from the skin. There is not a slide for that, but AEB just developed an enzyme which is called enzyme CAT0, which stands for catechin 0. So it's an enzyme that you can use as a pectinase, as a maceration enzyme to extract from the skins without having too much of a polyphenolic extraction, actually. So to finish the enzyme part. But then oxygen management. How can we minimize the oxygen pickup and the oxidation of this sulfur-containing components? These are old research that I am reporting here. One from Debardieu, former professor of enology at the University of Bordeaux. He spent many years researching glutathione in Sauvignon Blanc, and you can find many articles where basically he sustained that glutathione is always useful to keep Sauvignon Blanc on lees, because the lees contain glutathione that will prevent the color from yellowing, dissipation of the varietal aromas, and the wine tendency toward developing aging defects. Another professor that was talking about glutathione is Vernon Singleton at UC Davis, and here we're talking 1984. Is not regarding thiols. This is more about color, but once again, it was investigating the antioxidant effect of glutathione, which recently actually, compared to this research, probably around 5 years ago, glutathione was authorized as an ingredient by the OEV. And as much as we can't really go and buy from our supplier glutathione to add to fermentation, we can add products like élevage glue, which are yeast extracts with a very high content in glutathione. We can also use gallic tannin like gallium, which have a very high affinity for an oxygen and can guarantee a good level of protection from oxidation. So again, oxygen management should be also done when it's time to close the bottle. And this is a piece of equipment that AEB can supply through one of its controlled company called Innotech. It's a piece of equipment that is called Thiols, where there is a pump that collects the wine from the cellar. The oxygen is checked. It's a flow meter just to make sure, to know how much we need to adjust, because at this point we already set how much oxygen we want to go in the bottle. There's going to be nitrogen sparging to strip the oxygen in this tank, which is under pressure and under nitrogen. The pump will bring the wine without the oxygen forward if the level of oxygen is what we desire. If not, it will actually go back in the stripping tank, being stripped, and the machine will receive a message so that it can self-calibrate to make sure that it will act differently, because it learned that the oxygen level is not reaching the point that we set originally. So this can happen if the bottling line has a slow intake and there is a change of flow. The machine can recalibrate itself every time so that every bottle is the same. The gas is checked also at the end to make sure that there are no problem in the line, and eventually, if there are, the wine is sent back again for recalibration. There's a PLC that controls the machine, and finally, the line to the bottling. Another product that concerns aromatics is the Thermoplast Pure-Off. We decided to include this in the presentation because methoxypyrazine can be a big part of the varieties that we mentioned, including Sauvignon Blanc, but especially Cabernet Sauvignon and Merlot. Methoxypyrazine come from grapes, so they're not secondary but primary aromas. They have different formulation. The most common is the IBMP, the 2-methoxy-3-isobutyl pyrazine. Smells like green pepper. And then there are the others with the famous aromatics that are perceivable at very low levels. We're talking just a few drops in a pool as far as perception threshold. What can we do to eliminate this? Not much, unless we do, say, thermovinification, but that's not what we're going to do, especially if we want to enhance thiols and esters. We're going to try to absorb or mask this methoxypyrazine. Oak chips, yeast derivative, and AAB has selected a specific yeast membrane, very spongy, that can absorb this methoxypyrazine. We can use it in fermentation because it's a component of yeast, so it can go in as, say, a nutrient. Contains actually a little bit of amino acid, but his main goal is to absorb odors like pyrazine and eventually also smoke taint. So we added to the fermentation, even with some enzyme to facilitate the extraction eventually. And when the wine is racked, the pyrazine, a good chunk of the pyrazine are racked with the lees. So thank you. Have a great harvest 2022 for the Southern Hemisphere right now, in a few months for the Northern Hemisphere, and we'll see you for the next one.